So now we'll discuss DNA and RNA structure and analysis. The genetic material must exhibit four characteristics. Genetic material must be able to replicate. Second is it must store information to pass it to the next generation. It should express information and allow variation by mutation. This is the central dogma we have already discussed. DNA to RNA, DNA replicated, DNA to RNA transcription, and from RNA proteins are formed. The process is translation. Until 1944, observations favored proteins as genetic material. The genetic material is physically transmitted from parent to offspring. Proteins and nucleic acids were the major candidates for the genetic material. For a long time, proteins were favored to be the genetic material because of their abundance into the cell. That is, the protein concentration is highest into the cell out of 4 biomolecules. It was the subject of most active areas of genetic research. DNA was thought to be too simple to be the genetic material with only 4 nucleic acids as compared to the 20 different amino acids of proteins. DNA is found only where the primary genetic function occurs, whereas proteins are found throughout the cell and in all stages of life cycle. This provides indirect evidence for DNA as genetic material. UV light. UV light is absorbed by DNA as well as proteins. So at 260 nanometers, DNA and RNA absorb maximum UV light. Therefore, 260 nanometers is absorption maxima for nucleic acids, and at 280 nanometers, proteins absorb maximum UV light. So 280 nanometers is absorption maxima for proteins. Whenever you need to determine the concentration of DNA or RNA, the absorbance of solution containing nucleic acids is measured at 260 nanometers using spectrophotometer. In case of DNA, if the absorbance at 260 nanometer is equal to 1, exactly 1, then the concentration of DNA is 50 micrograms per ml. Okay, 50 micrograms per ml. And in case of RNA, if absorbance at 260 nanometers is equal to 1, it is 40 microgram per ml. Concentration of RNA is 40 micrograms per ml. Okay. For the proteins, two 80 nanometers is absorption maxima. The strongest evidence for DNA as genetic material came from recombinant DNA technology, from the use of recombinant DNA technology or genetic engineering. Segments of eukaryotic DNA corresponding specific genes are isolated, spliced together and inserted into the bacterial cell. The presence of eukaryotic gene product in bacteria containing the eukaryotic gene provides direct evidence that this DNA is present and functional in bacterial cell. Some viruses, they have RNA core, that is RNA as their genetic material rather than the DNA. This RNA serves as the genetic material for these viruses. For example, tobacco mosaic virus. If you can see here, these are the code proteins of tobacco mosaic virus, which helps in forming helical structure of this RNA. along with the coiling and condensation of this RNA. Okay. Replication of the viral RNA is dependent on RNA replicates. Retroviruses, this is the different class of virus, they replicate in unusual way. That is, they have RNA as their genetic material. From this RNA, double-stranded DNA copy is synthesized using special enzyme which is present in this retroviruses known as reverse transcriptase. Wait a minute. Okay, so this reverse transcriptase. Reverse transcriptase is RNA dependent DNA polymerase. Why? Because it use RNA as template and synthesize DNA from it. Okay, and this DNA is then incorporated into the host cell's genome. Now the DNA structure. Now DNA, as we have already discussed, contain nitrogenous base, sugar and phosphate group. Nucleotides are building blocks of the DNA. Nitrogenous bases are of two types, purines and pyrimidines. Purines are adenine and guanine. Pyrimidines are cytosine and thymine and uracil. This uracil is present only in RNA, and in RNA thymine is absent. So these are purine and pyrimidine rings. This is pyrimidine ring, this double ring structure is purine, and this one is ribose sugar. Okay. So this ribose sugar, if you see the carbon atoms, this is pentose sugar. Why pentose? Because it consists 5 carbons. This is carbon number 1, 2, 3, 4 and 5. These nitrogenous bases, they also have carbon atoms present. See this one, this one, this one. Here also in pyrimidine ring, this one along with the nitrogen. So if you see here in this purines and pyrimidines, number is given normal, like 1, 2, 3, 4, 5, 6. Here 7, 8, 9 also present in purines. And in this pentose sugar, ribose sugar, numbering of the carbon is done in this way: 1 prime, 2 prime, 3 prime, 4 prime and 5 prime. Only 5 carbons are present, therefore it is pentose sugar and it is a ribose sugar. Now here you can see at 2 prime and 3 prime hydroxyl groups are present. Here this is 2-deoxyribose, 2 prime deoxyribose. Here OH group is absent. See here, normally this is normal form ribose sugar where OH group is present. Here this oxygen is removed, therefore it is deoxygenated. So it is 2 prime deoxyribose sugar present in DNA molecule. Okay. Now DNA and RNA both contain ACG. Only DNA contains thymine along with A, C and G, and only RNA contains uracil along with A, C and G. Therefore the DNA contain A, T, G, C nucleotides, nucleic acid bases, and RNA have A, U, G and C. RNA contain ribose sugar and DNA contain deoxyribose sugar. Okay. Nucleoside and nucleotide. Now this is the nucleoside where sugar and this base, these two are linked to one another. Now this bond which is formed, which links these two, is glycosidic bond, this bond. And then to the fifth carbon of sugar, when phosphate group is attached, this one, it is nucleotide. Okay, this phosphate group is linked with the ester bond. Sugar and base are linked with the glycosidic bond. Okay, so ribonucleotides sides are adenosine, cytidine, guanosine, and ribonucleotides are adenyllic acid, cytidic acid, guanylic acid, and uridylic acid. Deoxyribonucleotides are deoxyadenosine, deoxycytidine, likewise, and deoxyribonucleotides are deoxyadenyllic acid, deoxycytidyllic acid, likewise. Now, on the basis of presence of phosphate group, how many phosphate groups are present, 1 or 2 or 3, decides what type of nucleotide it is, whether it is NMP. If only single phosphate group is present, nucleoside monophosphate, nucleotide diphosphate if 2 phosphate groups are present, and if 3 phosphate groups are attached, it is nucleoside triphosphate. Okay, 1 phosphate group, NMP, nucleotide monophosphate, 2 phosphate groups, nucleoside diphosphate, and 3 phosphate groups, nucleoside triphosphate. Now see, this one is NDP, deoxy NDP, and this is NTP. Here only 2 phosphate groups are attached, here 3 phosphate groups are attached. So it is ATP, adenosine triphosphate, and this is deoxythymidine diphosphate. Okay. Nucleotides are linked together with a phosphodiester bond. I'll show you how. Now see this one. See this one. Okay. Now these two, this is say thymine and this is adenine. Thymine and adenine. Sorry, thymine and guanine. Thymine and guanine. These two are adjacent nucleotides present in one strand of the DNA. Say this one is T and this one is G. These two are linked together with a phosphodiester bond. This bond which is present in between these two, this bond is known as phosphodiester bond, which is formed shown here. Now how these two are linked? See, the previous bases, say for example previous base is thymine. Now this thymine's 3' OH of deoxyribose sugar, or say simply sugar, is linked to incoming nucleotide 5' phosphate. 3' OH of the previous base is linked to the incoming or adjacent base 5' phosphate. This is how the phosphodiester bond forms. Okay, and this is how the bases are linked to one another. Here you can see the bases are shown in one strand, A, T, G, C and A. At 5' side you will always find a free phosphate group, and at 3' side free 3' OH, free 3' OH. This OH or this end of the DNA is always known as growing end, because new nucleotides complementary to the template strand are added or attached to this free 3' OH of the DNA. So this is known as growing end. Chargaff rule we have already discussed. Base pairs AT and GC are same. Amount of A is proportional to the T and amount of C is proportional to the G. Now this A and T in G and C, they are known as complementary bases. Always adenine in one strand pairs with the thymine. Say if we consider these two strands of the DNA, and these are the say bases present in two adjacent strands. If say adenine is present in one strand, it always pairs with the thymine in its adjacent strand. If cytosine is present here in one strand, it always pairs with the guanine in adjacent strand. So these two are known as complementary bases, A, T and G, C. And if you see the ratio, it is always equal to 1. AT and GC ratio always equals to 1. X-ray diffraction of the DNA. This is the X-ray diffraction structure studied by Rosalind Franklin. Helical structure. Watson and Crick proposed the DNA structure, complete DNA structure. Watson and Crick model of the DNA, which is right-handed helix, right-handed double helix. Two strands are anti-parallel and twisted, coiled around one another, and the bases are stacked on one another. Two strands are connected by AT and GC base pairing, and there are 10 base pairs per helical turn. Anti-parallel in the sense, see, if this is one strand of the DNA and this is its adjacent strand, one strand runs 5' to 3', other runs 3' to 5'. Bases are always complementary to one another. Now here you can see in one turn there are 10 bases. The distance between each base pair is 3.4 angstrom. This is one base pair and this is another base pair. Now see, here you can see blue colored one is adenine, this blue colored one is adenine, yellow is thymine, grey is guanine, pink is cytosine. So in between each base pair the distance is 3.4 angstrom. Now the two strands, see here you can see, are coiled around the central axis. This black line is central axis around which these two strands are coiled. Now where at some points you can see the angle of rotation is wider and here it is narrower. So the wider angle is major groove, termed as major groove, and narrower is minor groove. Okay. Now here you can see this pink colored backbone is formed by sugar, alternating sugar and phosphate, and its bases are embedded inside of the helix. They are stacked around one above the other. See here you can see this is backbone formed by this sugar, phosphate, and these are the bases which are embedded inside of the helix. Here you can see sp, sugar and phosphate. Okay. AT and GC base pairing provides complementarity of the two strands and chemical stability of the helix. AT base pairs form two hydrogen bonds, AC form two hydrogen bonds, and GC form three hydrogen bonds. Okay. See, here you can see these are 3 hydrogen bonds between G and C and 2 hydrogen bonds between A and T. Arrangement of sugars and bases along the axis provides another stabilizing factor. Now these are the different forms of the DNA: bDNA, aDNA and zDNA. The Watson and Crick model of the DNA is bDNA form, which is believed to be biologically significant and most stable form. Most common and significant right-handed helix, 20 angstrom diameter. Bases are complementary to one another, AT and GC. Each base pair has same distance, approximately 10.85 angstrom. So this is bDNA. Now what do you mean by right-handed helixes? Imagine your right hand, turn all the fingers inside and open up your thumb. So your thumb is an axis and your folded fingers, 4 fingers which you have folded to form a punch like boxing. Open up your thumb. These folded fingers, if you see, each finger is turning to the right side, that is in anti-clockwise manner. 2 strands are coiled around one another in anti-clockwise manner. So this is right-handed helix, and ulta in case of left-handed helix. Fold your fingers of left hand, open up your thumb. Now see your fingers. Each of your finger is turning in clockwise direction. This is how the 2 strands of the DNA are coiled around one another in a clockwise manner, and this thumb is, say suppose, the axis of your DNA. Now this bDNA contains 10 base pair per turn. Pitch is 34 angstrom. Pitch, that is helix per turn, base pairs per turn. Axis passes through the middle of each base pair. Minor groove is narrow, major groove is wide. A is also right-handed helix. Only the difference is 11.6 base pair per turn are present. This is wider and flatter than bDNA. Here you can make out the difference. This is bDNA and this is aDNA. aDNA is wider and flatter than the bDNA. 11.6 base pairs are present per turn. Major groove is deep and minor groove is shallow. Observe under dehydrating conditions. Okay. When relative humidity is approximately 75%, bDNA form is converted to aDNA form. The process is reversible. The most self-complementary oligonucleotides of 10 base pair crystallize in aDNA configuration. aDNA has been observed in 2 contexts: at active site of the DNA polymerase, in gram-positive bacteria undergoing sporulation. When cells are exposed to UV, that time also bDNA form is converted to aDNA form. Then the zDNA. zDNA is left-handed helix. 12 base pair per turn are present. Pitch is 44 degree. Pitch मतलब number of in one turn, what is the angle? Minor groove is deep, no discernible major groove. Reversible changes from bDNA to zDNA in localized regions may act as a switch to regulate gene expression. Now this zDNA is a left-handed helix seen in conditions of high salt concentration, mostly present in extremophiles. Okay, zDNA form you can observe mostly in extremophiles. In complementary polynucleotides with alternating purines and pyrimidines, G can pair with G, A can pair with G. So this is uncommon feature in extremophiles which have Z form of the DNA. Purines can pair with one another. Might also be seen in DNA segments with above characteristics. ADNA is slightly more compact, flatter than B. C, D and eDNA are also right-handed forms of the DNA, less compact than pDNA. Okay, so here it is the bDNA, this is aDNA which is flatter, more flatter and wider than the bDNA, and this one is zDNA which is zigzag form of the DNA. Zigzag and left-handed. Okay, zigzag and left-handed. Now next is the difference in DNA and RNA. DNA sugar present is deoxyribose. In RNA ribose sugar. The bases present in DNA are ATGC, here AUGC. This is the difference. Mostly DNA are double stranded, RNA are single stranded. Genetic material is mostly in all life forms is DNA. In some viruses you will find RNA as genetic material. DNA does not have any enzymatic activity. Many RNAs, they have enzymatic activity. They can cleave other RNAs or themselves, also known as ribozymes. Structure is double helix, and this RNA molecules are either linear or folded. Structure of RNA is chemically similar to DNA, but it is single stranded. Like this single stranded, but they can form intra-strand base pairing, that is base pairing formed within single strand of, say suppose this is single stranded RNA molecule. If it contain inverted repeats, that is say here G U G is present, here C A and C if present, these can form base pairing with one another, since these bases are complementary. G is complementary to C, U is complementary to A, and this G is complementary to this C. So therefore RNA can form intra-strand base pairing like this. Okay, and this structure is known as formation of hairpin like structure. Most RNAs are single stranded. Only few viruses they have double stranded DNA. Some RNAs form double stranded regions as they fold into different secondary structures. These are known as secondary structures. This is double stranded RNA virus. This is virus, HIV virus, which have RNA as its genetic material. Few viruses they have RNA as their genetic material, either present in single stranded form or in double stranded form. There are three major classes of RNA: mRNA, rRNA and tRNA. The characteristics of these RNAs in prokaryotic organisms and eukaryotic organisms are summarized in this table: mRNA, rRNA and tRNA. mRNA stands for messenger RNA, tRNA stands for transfer RNA, rRNA stands for ribosomal RNA. mRNA codes for protein, ribosomal rRNAs are used in translation for forming ribosomes. tRNA are also used for translation, which brings or drops amino acid during protein synthesis. yRNA is used for RNA processing and DNA replication. Telomerase RNA used for telomere synthesis. Regulatory RNA is known as antisense RNA, aRNA. These are used for transcriptional attenuation, mRNA degradation, mRNA stabilization and translation block in all organisms. This is the total percent of RNA. rRNAs are 80%, tRNAs are 50% of the total RNA and mRNA is 5% of the total RNA. These are their sedimentation coefficient, Swedberg units, and these are sizes. rRNA size varies in pro and eukaryotes. tRNA size is almost same, 75 to 90 nucleotides, and mRNA is 100 to 10,000 nucleotide long. All RNAs originate as complementary copies of one of the two strands of DNA. DNA serves as template for protein synthesis, RNA synthesis. rRNA are components of ribosomes. mRNA are templates for protein synthesis, and tRNA carry amino acids for protein synthesis. Now these are analytical techniques which have been useful during the investigation of DNA and RNA. Nucleic acids, they absorb maximum UV light at 260 nanometers due to interaction between UV light and the ring systems of the bases. Purine and pyrimidine rings, they absorb this UV light. Nucleic acids can be separated by gradient centrifugation technique. Sedimentation equilibrium centrifugation separates by density gradient, and sedimentation velocity centrifugation measures velocity of sedimentation in Swedberg coefficient units. Now this is the picture of density gradient centrifugation. This is the first stage. sample of nucleic acid loaded onto the top of concentration gradient. This is density gradient which is already formed using either cesium chloride or fecol or sucrose. Now the density gradient forms means the density of the solution is heavier at the bottom, decreasing towards the top. ये जो blue solution you can see, this has highest density at the bottom, decreasing towards the top side, and at the top side density is lowest. So on this density gradient, solution of the DNA is loaded, or solution sample to be separated according to the density is loaded over the top of this density gradient. Then these tubes are centrifuged. Okay, once centrifuge, the sample, they run down the density gradient and sample is immobilized where the density of the solution, this solution, matches with the density of sample present in this solution. Okay, you can see here two colored bands, one is red, one is yellow. Okay, now these two colored bands means this is the mixture of nucleic acid separated, because these two nucleic acid segments, they have different densities. Okay, so they are immobilized at the point where their density matches with the density of solution. Okay, then the samples are eluted out. See, this yellow is separated in 6th tube and this red will be separated later in 9th tube, and then they are absorbance is measured using UV spectrophotometer at 260s. Hyperchromic shift during the DNA denaturation is used to determine melting temperature. Okay, melting temperature is the method or denaturation method for estimating base composition of the DNA. This is melting temperature. Now see this DNA molecule. Here only one DNA molecule is shown. If the solution containing DNA molecule is heated, see at previous the temperature is say less, 10 to 20. As you go on heating this solution, these two DNA strands which are coiled around one another, these will separate from each other by breaking hydrogen bonds present between these two DNA strands, between say AT and GC base pairs, due to higher temperatures. And at last stage somewhere you will find two separate DNA strands, single strands of the DNA. So Tm is the temperature at which half of the DNA is denatured. Okay, half of the DNA is denatured, is known as T. Okay, so this effect is known as hyperchromic effect. Hyperchromic effect, as you separate these two DNA strands from one another, if you take absorption at this stage where the DNA is present in intact form, you will find less absorption. See here you can see this is near to 0. Okay, say for example 0.1. As you go on heating this DNA solution, you will get two single strands of this DNA separated because of higher temperature. This process is known as conversion of this double-stranded DNA to this single-stranded form is known as denaturation or melting of the DNA, which occurs simply because hydrogen bonds, they break and then two strands, they separate from one another. If you take absorption at this stage, you will find higher absorbance. देखो यहाँ पे it is almost 0.1. Here now it is almost 0.75 or 0.8 something. Okay, so this absorption increases, therefore this is known as hyperchromic effect. Okay, reverse case is also possible. Now here you have two separated single strands. Now when you allow the same solution to cool, again after some time you will get double stranded DNA. Now each of this strand will find its match. It will try to find its match by pairing with complementary bases. Then double stranded DNA is formed at the cool down the solution. Then again the absorption decreases, OD decreases. This reverse case from here to here, when absorption decreases, is known as hypochromic effect. It decreases, hypochromic effect. Molecular hybridization, well, the DNA and RNA strands can be renatured to each other. This provides basis for several techniques for analyzing DNA. See this picture, what I was telling you, the here three strands are shown, number 1, number 2 and number 3. Now as you heat this solution, you will find now here you will find 6 separated single stranded DNA molecules. As these hydrogen bonds are broken, since initially there were 3 double stranded DNA molecules, they are converted to 6 single strands of DNA. Then this RNA transcript complementary to one DNA strand is added here. Okay, this is complementary to DNA strand of number 3. Now DNA and RNA are mixed together. Now since this black colored one is complementary to this red one, this can form either double strand with this molecule or this RNA transcript. Okay, the complementary duplexes form, including this DNA RNA hybrid. So this process is known as denaturation. Hypochromic effect it shows, that is optical density will increase. This is known as renaturation or re-annealing. It will show hypochromic effect. Here you will find higher absorbance at 260 nanometers. Here at this stage, when two double stranded DNAs are formed, absorption will decrease. Now why this absorption increases and decreases? See this double stranded DNA, what absorb light, UV light, are nucleic acid nucleotides, which are embedded here inside of the helix. They are not exposed out. As these molecules are converted to single stranded forms, the bases are exposed and they absorb maximum UV light, and therefore it will show higher absorbance. Now when again single stranded DNAs are converted to double stranded form, absorbance again decreases. Since all the bases, they are now again embedded inside of the helix, they will not absorb maximum UV light. Okay, so absorbance again decreases. This is hypochromic effect. The next technique is fish fluorescent in situ hybridization. In situ means in that particular organelle, or say in that particular system, in situ, is used for identifying the chromosomal location of the DNA of interest. See, here you can see these are the chromosomes, and these yellow colored are the primers which are attached to the specific for say centromere. Reassociation kinetics we have already discussed, provide information about the size and the complexity of genomic DNA from an organism. Cot curve is studied. Then the electrophoresis separates DNA and RNA fragments by size such that smaller fragments migrate through the gel faster than the larger fragments. So this is the gel which is formed. This is the gel which is formed. These are agarose units, black circles which you can see. Here when you load the DNA sample, now this DNA fragments are of variable sizes, है ना. छोटा वाला होगा उसका size और molecular weight is less than this larger DNA fragments. As you load it and provide power supply, all the molecules, since DNA is negatively charged, they travel towards the positive charge. Smaller the molecular weight, less the molecular weight, faster it will travel from the spaces which are available in between these gel particles. Heavier the molecule, it will take time to travel. Okay, so here you can see सबसे small जो होगा that shows band at the bottom, and the largest one, the heavier one, the one which has highest molecular weight, shows band near the well. Okay. So that's what about the DNA and RNA. Thank you.